pleasure to introduce the speaker of today's seminar. It is Koen Verhoeven. I will surely make wrong in pronouncing your name, but actually I'm very glad he decided to come here. He works actually at the Netherlands Institute of Ecology in Wageningen, and this is an institute of research like the Stazione Zoologica, but devoted, it's not university, it's devoted to the study on ecology, and Koen works on terrestrial plants. He got his PhD at the same institute, but since then working actually on the genetic of local adaptation in wild barley, working with the quantitative trait loci, the QTL. But then he moved to US for the postdoc in Indiana, where he started to work on computational genetics. Then back to the Netherlands, where he got to become interested in epigenetics and in the use of epigenetic approaches for studying wild populations of terrestrial plants. And the Tarassacum is becoming now his pet species. So I think that's enough to introduce him. We are very anxious to listen to your seminar, and the title is Epigenetics in the Wild, Causes and Consequences of Natural Epigenetic Variation. Thank you.
Does this work? Yes, this works. All right, second try. Could you hear me, by the way? You're not with. Okay. So this species, a well-known flower polymorphism that was known for a long time. And when people started to, it was a very stable mutation. People consider this a normal mutant. It's segregated in crosses. It behaved like any other mutant. But people found that in some crosses, the mutant phenotype could revert to the wild-type phenotype. And also in individual plants, some branches could have mutant flowers and normal flowers. So when people started looking into the molecular basis of this, what they found was that this was an epigenetic mutant. So they found a gene involved in flower morphology and development of flower morphology. And they found that there was, in fact, no relevant sequence variation between the two flower morphs, but what was consistently different between the different morphs was the DNA methylation status of the allele, of the two alleles. So there was a methylated, let's say a methylated variant of the allele and an unmethylated variant of the allele, and that associated 100% with the phenotype. And they could even see that in cases where the phenotype reverted from mutant to wild type, that was associated with a shift back from one methylation status to the other. So this is an old study, 1999. But it showed that even in the absence of genetic variation, you could have heritable phenotypes and potentially relevant heritable phenotypes out there in nature that are determined not by DNA sequence variation, but by epigenetic variation, in this case DNA methylation, which is the main epigenetic mechanism that I will talk about today. So DNA methylation, so you look at the chromosome, and we all know, so the information is encoded in the DNA sequence, but how the DNA sequence is made available for functional, for enzymes, it's determined by histone modifications and also DNA methylation variants. So these are chemical, stable but reversible modifications of the DNA sequence. So in this case, for methylation, these are methyl groups added to cytosines in the DNA sequence. They're relatively stable. Stable means these DNA methylation, it is heritable from cell division to cell division. And as we will see, part of it in some cases is so stable that it is heritable from generation to generation even. So there's many things in plant genomes, many things about DNA methylation that we do not completely understand, but at least one function of it is the regulation of expression. So regions in the genome that are densely methylated can suppress gene activity, and this is particularly clear in plants for the silencing of transposable elements. So in plant genomes, transposable elements are suppressed by DNA methylation. And if DNA methylation, dense DNA methylation occurs in the promoters or genes or around the transcription start site of genes, this is also associated with reduced gene expression. Having said that, and we will see a bit later, more about that a bit later, there's also a lot of DNA methylation in the genome that we actually do not know what the function is, and it doesn't always associate with gene expression. As I said, it's stable across cell divisions, but also in some cases across, through meiosis across generations. So giving potential for transgenerational epigenetic inheritance, affecting gene expression states in a stable way across environments. We also know that some, like most of the epigenetic mechanisms, so you have histone modifications, DNA methylations, you can think of other mechanisms that people consider epigenetic mechanisms. Often those are sensitive to environments, and one idea is that they help adjust the genome to be translated into a specific transcriptome to affect gene expression levels to the environment. So with these things known, there's been a lot of speculation. Okay, so what can these stable gene regulation mechanisms mean for ecology and evolution? So people started looking, like I did, to DNA methylation in, for instance, ecological context. And it's very important to realize right from the start that DNA methylation is a mixed bag. So what this means, as we'll see in the end, is that it's going to be necessary, I think, to do detailed molecular analysis on DNA methylation in species where you can actually look in some detail into the molecular, into the DNA. So at some point, I will argue you will need a species with a reference genome to do high quality epigenetic work. And part of that is because DNA methylation, so this is in plants, in animals it's a bit different, but in plants we see, for instance, that methylation is always on cytosines, but the cytosine can be in different sequence contexts. It can be in CG context, so sitting next to a G, or sitting next to something else and then a G, or just any cytosine in the genome can be methylated. But if we look at plant genomes, we see an overall landscape where transposable elements are often heavily methylated in all sequence contexts of cytosines. In gene bodies, we mostly see methylation in plants in CG context, and it turns out that these different types of methylation, they're very different in different aspects. So they occur in different sequence contexts and in different genomic contexts, but there's also completely different enzymatic pathways involved in depositing the DNA methylation, maintaining it across cell divisions, and selectively removing it again. So these are different types of DNA methylation. They have different functions, so this type of DNA methylation that is very dense over transposable elements or maybe in nearby genes, it can really affect gene expression. But there's a lot of DNA methylation, for instance, in CG context, in plant genomes, in gene bodies, that simply does not associate with gene expression or not very well. So we don't really know what the function is. It may have to do with hiding hidden transcription start sites, or it can have to do with splice variants. There's all kinds of ideas, but it's for sure not always correlated with gene expression. The different types also have different heritabilities. In plants, again, this is different between some plants and animals, but in plants we know that DNA methylation in CG context mostly is simply not reset in germlines or during meiosis, so it stably travels from generation to generation. It's not erased and then rebuilt again. It actually continues to sit on the DNA as far as we know. But this is different for DNA methylation in the other context. And also the different contexts seem to have different environmental sensitivities. So I wanted to point this out from the start. So it's one thing to look at DNA methylation in the genome, but you're looking at everything at the same time. Lots of it in plants is just sitting on transposable elements and doing nothing for most of the time. Suppressing the expression of the transposable elements, but not really sensitive to environments, for instance. So to really understand what is going on with DNA methylation in your system, actually, in the end, it's going to be important to be able to look at this level of resolution of DNA methylation. So I say that now at the start. Then, of course, I entered this field as an ecologist, looking at DNA methylation in a non-model species, where we simply ignored all of these details, and we simply were happy to look at some DNA methylation in the genome and see if we could infer something of that. So what I'm going to do in the rest of this talk is I'm going to spend a few more moments on thinking about conceptually what are the potential contributions of epigenetics in ecology and evolution. Then I'll show you some of the results that I had in my dandelion system, where actually measuring DNA methylation is something that only now we are starting to be able to do in some level of detail. And I will actually mention something of the methods that we used, that we developed and used for measuring DNA methylation in non-model systems. So very quickly, some conceptual background. So if you think about epigenetics and evolution, why is it interesting? So where is a potential contribution? So what I draw here is simply, maybe you can even think away epigenetic variation, and if you would just think of phenotypic variation, then you have the classic system of how plants and animals deal with different environments and adaptation to environments. So at the population level, you have Darwinian evolution through selection on random genetic variants, heritable variants, shaping population level adaptation through a Darwinian process. And at the level of the individual, you have phenotypic plasticity. So depending on the environment in which the individual develops, the phenotype can be adjusted to the environment, and through phenotypic plasticity. And that in itself, of course, can be that's under genetic control as well. That's an adaptation in itself. So there's for instance, G by E interaction. Different genotypes have different abilities to be plastic. So this is, I would say, the basic default standard situation for thinking about epigenetics. But then with epigenetics, so there is I would say, one of the promises of epigenetics is that there are some superimposed on this basic model. It's back on, no? Yeah, okay. I'll try not to touch this. So there's an error here that suddenly becomes possible, at least in theory, with epigenetics, if it is the case that epigenetic modifications are triggered by an environment, and if they're so stable that they can be inherited, then you can have phenotypic plasticity being carried over a generation, or maybe 2 generations, or 3. I mean, this is an empirical question to what extent this happens, but this is one of the promises of epigenetics, that adds some richness to the repertoire of phenotypic plasticity. I'm not going to show this, tell this, because I will run out of time in the end. But that was just to show that there is some evidence for that from Arabidopsis studies. I'll say something anyway. So the current status of evidence is that this idea is, in sexual plants, is that yes, this can happen, but it happens, it's not abundant, it's not a ubiquitous thing. So most of the epigenetic changes that are induced by an environment are reset within the same generation. A very small part of it is indeed still found in the offspring generation, where it affects gene expression and traits. But in Arabidopsis, you really have to look for those sites, and then the generation after that, they're gone again. So the phenotypically plastic response for a tiny part, for a small part, carries over to the next generation, and then it's gone. We see now in Arabidopsis that if you trick Arabidopsis to be parthenogenetic, so to avoid sexual sexual reproduction, then the induced epigenetic variants are stable. More of them are stable across generations and for more than one generation. So avoiding the germline process and the sexual reproduction process is favorable for the stability of epigenetic variation. So that was what I call the phenotypic plasticity domain. So to me, a completely different domain that has nothing to do with environmental induction is this process of Darwinian selection, where epigenetics is relevant because you can have epigenetic mutations build up, just like genetic mutations build up in genomes, that are, after they arise, may be stable across generations. And I showed you something about the landscape of methylation in plants. We know now in plants that DNA methylation in CG context, in gene bodies, if a change occurs, an epigenetic status in that context, then it's really stable over many, many generations. This can be inherited. The issue there is that we don't really know what the function of that DNA methylation is. But potentially, as we saw in the first Linaria flower example, there can be epigenetic mutations that are just sitting there, really stable. Maybe not so stable as genetic mutants, but much more than 1 or 2 or 3 generations. That if they happen to affect the phenotype, then that phenotype, the heritable variation in that phenotype, is not only encoded in the DNA sequence anymore, but part of it is encoded in the epigenetic code, which is then visible to selection. So part of the selection response can be based, in that case, on epigenetic variants and not on DNA sequence variants. So this loop is also an added possibility for adaptation with epigenetics. And just a hint of the potential of this is a study that they've done in Arabidopsis. So Arabidopsis is a completely inbred homozygous sexual species. So you can take offspring that are all genetically identical to the mother plant, and they maintained mutation accumulation lines for 30 generations, initially simply to see how genetic mutations build up in such a system. But because this experiment was available, people have also looked at the DNA methylation variation that builds up. And you see a result of that study here. So across if you take the generational distance between any two plants, so for instance, a plant here is 60 generations removed from a plant here. And what they plotted here is the divergence, the overall divergence genome-wide in DNA methylation in CG context, but CG context split out to various genomic contexts, so within gene bodies, within transposable elements, within promoters. Then what they see that for instance, in these green points in gene bodies, that really the divergence between two individuals becomes more and more bigger and bigger and bigger with more generational distance. So this is simply building up of stably inherited epigenetic mutants. Just like over longer generations, genetic variation and divergence would build up with increasing generations. But this really also happens with epigenetics, with DNA methylation. So it differs a lot between the contexts. So for transposable elements, there is some divergence building up, but very little. And this is because transposable elements, if methylation is lost there, the plant has a system in place to actively restore it. So there's less scope for diverging over time between lineages. But some DNA methylation clearly is very dynamic, and it builds up, and it sits, and it continues. So with that conceptual picture in our minds, we moved to the studies, some of the studies that I've done in asexual plants. So I figured, so with sexual plants, there is some scope for transgenerational stability, but we see the environmental induced part of it is inherited maybe for one generation and much is reset. But I figured plants that circumvent sexual reproduction also circumvent the epigenetic resetting mechanisms that take place during sexual reproduction. So I figured that maybe those are cool systems to look at epigenetic variation that build up in those asexual lineages. And also because you can argue or you can hypothesize that in such systems, epigenetic variation will be a relatively important contributor to variation in the system compared to sexual lineages or sexual species. Because in asexual species, if everybody has the same genome, there's limited opportunity for adaptation, really. So I started looking, actually I forgot to put it in the slide to introduce the dandelion system. I started looking in these dandelion plants. So dandelions are plants that within the same species you have sexual plants that are outcrossing diploid species. And from sexual populations with a certain frequency, asexual parthenogenetic genotypes arise that actually have a higher ploidy level. They're triploids. It's not facultative. Once they're triploids, they're always parthenogenetic. So each parthenogenetic triploid individual that is produced in populations is leading its own evolutionary life from then on. So that's from then on, it's an asexual lineage. And this process of the generation of triploid parthenogenetic plants, this occurs frequently. So it's a system with huge clonal diversity. So there's literally thousands, tens of thousands, different clonal dandelion lineages co-occurring in Europe. And the lucky coincidence is that some of them, there's a few experts in the world, and some of these clonal lineages, individual lineages, can be recognized by 2 or 3 experts in the field. So people collected for me material from different countries of plants belonging to the same clonal or asexual lineage. And so I didn't believe, to me they're all dandelions, so when they said they could do that, I didn't believe them. So they sent me a box of seeds and we genotyped them, and they were almost spot on. They gave me 140 samples and 2 were misidentifications. And apart from that, they were spot on. So I have, for these studies, I have plants belonging to the same clonal lineage. And what we did, we grew them for a few generations in the lab to reduce some maternal effects. And then we saw that we grew them in a common experiment, and we saw that there's very significant heritable differences in the phenotypes of these plants. So here, for instance, flowering time, one of the most important traits for plants, was very different between accessions of the same lineage, depending on where they came from. They would flower either earlier or later. And so I was interested, so that this in itself is not indication that epigenetics is involved, but I was simply interested to what extent there was evidence that epigenetic variation that may have evolved within this lineage.
Maybe the battery. You can try the one that is on the podium. You need to turn it on.
Okay, yeah, okay. So as I said, this in itself is not, doesn't mean that there's epigenetics involved, but I was interested in that possibility anyway. So what I did is I evaluated if the flowering time differences between accessions were sensitive to a demethylation treatment. So in plants you can use in vivo treatments with chemicals that remove DNA methylation from the genome. So this is not a perfect method. If you use too high levels, then it becomes toxic, and it doesn't remove 100% of the DNA methylation. We know it removes maybe 30% or so of DNA methylation. But still, I performed this experiment where I thought, well, if these flowering time differences between accessions are caused by DNA methylation, then I should be able to make the flowering time differences go away if I make DNA methylation differences go away between the accessions. So I tried to do that using this Zeppelin treatment with removed DNA methylation. And lo and behold, I found exactly what I predicted. So these are cumulative flowering time distributions. I repeated the experiment with 6 different accessions that showed different flowering time in the first experiment. This is the control situation. This is also almost significantly different. I actually repeated this experiment in a different environment, slightly different with a cold period. And then this was highly significant. So this repeated what we know. In the normal conditions, these accessions show differences in flowering time. But with increasing methylation inhibitors during germination, the flowering time differences went away. So this was not a toxic effect. It was not that these plants just performed bad. We could not find any other phenotypic differences in these plants. And also the flowering was not all delayed or advanced. The only effect that we saw is that the flowering was synchronized. So this means that somehow the flowering time differences depended on the DNA methylation differences. So to me, this was an interesting result. It sort of shifts the question. So where do these DNA methylation differences come from in the first place? And we can only answer that question if we can look into more detail at the methylation status of the relevant loci here. So it can be that the methylation variation is simply spontaneous epimutations that are not controlled by genetics at all. But it's also a likely explanation that there are somehow genetic differences evolved within the clone of the asexual lineage that then, as a downstream consequence, controls the DNA methylation differences that control the flowering time differences. But this is a question that we can only answer by looking in more detail. I'm skipping this. One hypothesis is this, and this is actually what I would put my money on, is that during the evolution of this asexual lineage, transposable element mutations have occurred. We know that they're quite dynamic within such lineages. The slide that I skipped showed that there was quite some activity in transposable elements within a lineage. So what I think has probably happened is that within the lineage, a transposition event, so the jumping of a transposable element within the genome, may have caused a novel transposable element really close to a gene affecting flowering time. And if that new transposable element is then silenced by DNA methylation, which is done because the plant has this mechanism in place to do so, what we often see is that, at least in Arabidopsis, is that transposable elements that are really close to genes, and if they're silenced by DNA methylation, then the DNA methylation sort of spills over to the flanking regions and can also then silence the neighboring gene through DNA methylation. So it can be that this is the normal situation in the clonal lineage, that some plants don't have this insert and other plants do, and that affects the methylation status of a flowering time gene. And then if you force the methylation to go away, then everybody has the same expression level again. Whereas in the normal situation, you would see expression differences. So this could be one of the underlying mechanisms explaining the methylation effect on flowering time in the lineage. One other set of experiments that I've done is related to this plasticity component of epigenetics. So I was interested in whether in these asexual parthenogenetic plants, what happens to the epigenome if you stress plants and then take the stress away and follow the subsequent generations? Is something in the subsequent generations still affected by the stress treatment 1 or 2 generations ago? And I've looked at that at different levels, at the level of DNA methylation very crudely, at the level of gene expression, at the level of small RNA expressions, which can also play a role in the gene regulation, and which can also be inherited through a DNA methylation intermediate. So I did a bunch of experiments, and just to show one at the phenotypic level, at the gene expression level, we performed an experiment where we exposed plants for actually 2 subsequent generations to jasmonic acid, which it's a plant hormone, and it's involved in the defense signaling against herbivory often. So instead of doing an herbivory experiment, you can rub jasmonic acid on the leaves of these plants, and that sets in motion the defense response to herbivory. So we did that in the parental generations, and then we took offspring from treated and untreated plants, and then we performed a real experiment exposing each group to a common control environment, or also again to jasmonic acid. And we looked at, for instance, the RNA-seq gene expression to see if in these groups there was still an effect on gene expression detectable not due to the jasmonic acid treatment in the plants themselves, but the jasmonic acid treatment in the previous generations. So these red arrows, these comparisons, they give you let's say the direct effect of jasmonic acid treatment, and these blue comparisons track the parental or the grandparental effect of jasmonic acid treatment. So this was RNA-seq in a non-model species. So so there are some limitations to the analysis, but we found a couple of hundred genes, transcripts, genes that showed a direct jasmonic acid effect. There were about 500, and compared to those 500, we found a smaller number, but I would argue still a considerable number of transcripts that were differentially expressed not due to the jasmonic acid treatment of the plants themselves, but due to the jasmonic acid treatment that happened in previous generations. So yes, part of the effect carries over to the offspring generation. In the same material, we did not only look at gene expression, we also looked at leaf chemistry. This is we're looking at the defense response pathway, so the phenotypic effect may be expressed in plants' secondary chemistry. So we did LCMS metabolomics, so untargeted metabolomics. And if you look at the effects of the direct effect of jasmonic acid treatment, this is left to right. This is of course quite strong, but there was also a more subtle, a smaller effect, just like in the gene expression of the parental effect. So the leaf chemistry of plants was a bit different because of the jasmonic acid treatment that the parents of the plant had received. Functionally, so it turned out that the gene expression and the chemical analysis were really consistent in the results. So if you looked at the RNA-seq data, there were 92 genes that were differentially at a differential parental effect. We could annotate 20, and 2 of them hit the same enzymatic pathway, so an inositol phosphate kinase pathway. And these are very basic pathways, but are often involved in stress response, signaling of stress response. In the metabolomics analysis, we identified 31 compounds that showed a significant parental effect. So annotation in metabolomics is much more difficult than in gene expression. So it was quite some work, and in the end, only 2 of those could be annotated, and one of the 2 annotated ones matched the exact same pathway that was indicated by the gene expression. So this is functional evidence, both from gene expression and from chemistry, that there's a specific pathway that is hit, or that remains active in the offspring due to the effects of the environment of the parents. So, of course, we are interested. So this is what I showed is phenotypes, so chemical, molecular phenotypes, and we don't know if this is in fact epigenetics or DNA methylation causing this. And here we enter I think the main problem of epigenetic analysis in plants and in non-model species in general. So we want to address the question: are these effects mediated by DNA methylation? We get some crude insight in that by the DNA in the methylation inhibitor study, but actually looking in the genome is what you want to do, and this is of course difficult if you work in a species that doesn't have a good reference genome, where things are limited. So we invested. So there's ways of looking at DNA methylation in non-model species. And so we actually had performed an experiment that I'll show some results about later, where the same asexual dandelion genotype, we maintained replicate lines under control environments and under different environments, where we first had 3 generations of this jasmonic acid treatment, and then followed by 3 generations of controls. And looking at DNA methylation, what you really want to do, so if this was an experiment in Arabidopsis, the way to look at DNA methylation is to do a bisulfite conversion treatment. So bisulfite is a chemical that modifies cytosines in the genome and converts it to, in the end, Ts. They read as Ts after PCR. And it does that for normal cytosines, but not for methylated cytosines. So this is the basis after sequencing for distinguishing and recognizing methylated from unmethylated cytosines. And what you want to do, so again, if this was a species with a small genome that was referenced, you would do this bisulfite treatment, and you would sequence the whole genome with this treatment. This would be whole genome bisulfite treatment. And you could look at each cytosine in the genome, if it was methylated or not, or in more detail, in the population of cells, what was the level of methylation at that cytosine. It's often not 100%, but part of the cells will be methylated at that position and part not. So it's a quantitative measure. But if you do whole genome bisulfite sequencing, and you do this treatment, you align to the reference, and then you can compare, for instance, plants in different conditions, and you can simply see where are the methylation differences in the genome between the samples. And again, in the end, this is important because we know DNA methylation is not one thing. Here you can see what is the sequence context, what is the genomic context, etc. So this is what you want. You cannot do that if you work in a species that doesn't have a reference genome. Also, it's hard to do that if you work in a species with a big genome, because it's expensive, and our people do this in Arabidopsis, but Arabidopsis has a really tiny genome for plants, so that makes it also affordable. So what people refer to is reduced representation bisulfite sequencing, where they first select small parts of the genome associated with specific enzymatic restriction sites. So this is almost what old-fashioned AFLP was based on, and what genotyping by sequencing is based on, and red sequencing. So you look at the reduced representation of the genome, and maybe that's only 1% or maybe 0.5% of the genome that you can look at. And in those regions, you do a bisulfite treatment, and you look at the genome. And you could